Welcome to The Explainer. Today, we're zooming in, literally, on one of the most incredible fields in medical science. Think of your body as a massive, bustling city, and inside it, billions of microscopic citizens, your cells, are just going about their daily lives. But what happens when something goes wrong? When a disease quietly sneaks past the city limits? Well, medical science treats this exactly like a crime scene, relying on a brilliant form of cellular forensics to catch the culprit before it's too late.
Let's kick things off with a number that is just absolutely staggering. We're not just talking about abstract science here today. We're talking about a single breakthrough in this field of cellular forensics that has saved millions of women's lives all around the globe. No exaggeration. And how did it do that? By finding the microscopic fingerprints of cancer way before it ever has the chance to strike.
So, how did they do it? Well, it all comes down to cytology. If we break it down, cyto comes from Greek meaning cell, and logos means study. So simply put, it's the scientific study of cells we get from tissues or body secretions to pinpoint disease. Here's the core idea. Whenever your body goes through something, a disease, an infection, or even just a totally normal hormonal shift, it fundamentally changes the morphology, or the physical shape, of your cells. It's kind of like a suspect leaving a fingerprint at a scene. The cells literally change their appearance to tell us a story.
Here's how we're going to break it all down today. 1. The science of cells. 2. A life-saving pioneer. 3. Collecting the clues. 4. Upgrading the Pap test. And 5. Beyond cancer detection.
All right, let's jump straight into Section 1, the science of cells, specifically diagnostic cytology.
So the whole foundation of this field is studying those tiny structural changes. We call this diagnostic cytology, or cytopathology. It's all about morphology, the shape and structure of the cell. Think of a healthy cell like a perfectly round, plump water balloon. But if a virus or a cancer takes hold, that balloon might get jagged or swollen, or its internal nucleus might get super dark and distorted. Cytologists are trained detectives, literally scanning these slides to spot those weird, shape-shifting cells hiding right there among the healthy ones.
Moving right along to Section 2, let's meet the life-saving pioneer himself, Dr. George Papanikolaou. Take a look at the face behind the discovery. He was a Greek physician and anatomist working in the United States. And you know, if that name sounds a bit familiar to you, it's probably because he's the genius who introduced the life-saving Pap smear. Yep, named directly after him. This was an incredibly elegant, simple technique that allowed doctors to spot cervical cancer and other premalignant conditions way early enough to actually save the patient.
Looking at his timeline, he was born back in 1883, but the real game changer happened in 1928. That's the year he presented a groundbreaking paper called New Cancer Diagnosis, and in it, he proved something totally revolutionary, that you could reliably diagnose uterine cancer just by examining a vaginal smear under a microscope. Wild, right? By the time he passed away in 1962, this one technique had already completely transformed women's healthcare all over the world.
Okay, on to Section 3, collecting the clues. Let's talk cell sampling techniques.
So you might be wondering, how do we actually get these microscopic witnesses out of the human body and onto a glass slide? Well, scientists have come up with a few specialized ways depending on where the clues are hiding. The field is essentially divided into 4 main branches based on the sampling technique: exfoliative, abrasive, aspiration, and hormonal cytology. Each one uses its own specific set of tools and logic to safely extract that cellular evidence.
Let's zoom in on exfoliative cytology first. This one's really cool because it relies on what the body is doing naturally anyway. Cells constantly and spontaneously shed from the linings of our organs into our body cavities. It's kind of like how trees shed their leaves in the fall. Doctors can either collect these fallen leaves completely naturally, like checking cells shed in urine or sputum, or they can help things along just a bit by collecting the fluid with a syringe or a simple cotton swab, like they do in a vaginal smear. You can see exactly how a swab is used to collect the cells, and then comes the absolutely crucial step, carefully smearing that collection onto a glass slide. Once it's smeared, they hit it with a quick fixative spray. That instantly locks the cell's morphology in place, meaning they won't degrade while they're transported to the lab for the detectives to analyze. But what if the surface we need to look at doesn't just hand over its cells so easily? That is exactly where abrasive cytology comes in. Instead of just waiting around for cells to fall off, doctors get in there and grab them directly from the target surface, using scraping, brushing, or washing.
A classic example is the cervical scraper, introduced by Ayer back in 1947. Ayer's spatula lets doctors gently but firmly scrape cells right off superficial or deep lesions on the cervix, ensuring they get a really highly targeted sample.
Which brings us to Section 4, upgrading the Pap test with liquid-based cytology.
Now, science never stands still. Even Dr. Papanicolaou's absolute miracle of a test needed a modern upgrade for the 21st century. For decades, the traditional Pap smear was the gold standard, but it definitely had a flaw. Research confirmed the old Pap smear had an intrinsic sensitivity of only 51% for detecting cervical cancer. Think about that. That meant nearly half the time early signs could be totally missed, usually because the cells on the slide were all clumped together, dried out, or hidden by blood or mucus. So to fix this, in the early 1990s, medical engineers introduced a massive replacement, liquid-based cytology, or LBC.
And the impact of LBC? Immediate and profound. Just by switching to this new method, clinics saw a 65% increase in the detection of cancerous lesions. That is a real game changer. We went from essentially a coin flip chance of detection to a highly precise, super sensitive system.
This visual explains exactly why LBC is just so much better. Instead of smearing the cells right onto a slide where they can clump all up, the doctor takes the collection brush and rinses it completely into a little vial of preservation liquid. Then, in the lab, a machine spins and filters that liquid. It pulls out all the stuff that gets in the way, like the blood and mucus, and transfers just a single, perfectly clear, representative layer of cells right onto the slide. The cytologist gets a crystal clear picture of the crime scene.
Finally, Section 5, Beyond Cancer Detection, looking at its versatile applications.
Because here's the thing, cytology isn't just about hunting for cancer. Our cells react to basically everything, which makes this tool incredibly versatile. For instance, by checking vaginal smears, doctors can actually assess a patient's hormonal status. They track estrogen and progesterone levels to monitor pregnancies, look into sterility issues, or diagnose endocrine disorders. On top of that, cytology is right on the front lines for diagnosing different types of infections in the genital, respiratory, and urinary tracts. They even use it to carefully track how well a patient is responding to heavy-duty treatments like chemotherapy.
Oh, and here is a particularly fascinating application: sex determination. Doctors can take a really simple scraping of the buccal mucosa, which, by the way, is just a fancy medical term for the inside of your cheek, and look at the cell nuclei under a microscope. They're specifically hunting for something called a Barr body. It's a dense structure of sex chromatin that you uniquely see only in the nucleus of a female cell. So literally, just from a quick cheek swab, these cells instantly reveal some pretty deep genetic truths.
It's honestly amazing to think about. From Dr. Papanicolaou peering through his microscope back in 1928, all the way to the high-tech liquid-based processors we use today, diagnostic cytology has completely transformed how we read the microscopic language of our own bodies. Every single cell we shed from our skin, our lungs, or our organs holds this massive library of information about our health, our hormones, and our genetics. Which really leaves us with one thrilling question to think about. As our microscopes get sharper and our tech gets smarter, what other secrets are our cells just waiting to tell us? Thanks so much for joining me on this explainer. Keep questioning, keep learning, and I'll see you next time.